Neurotransmitters. Chapter 141. Definition. Neurotransmitter is a chemical substance that acts as a mediator for the transmission of nerve impulse from one neuron to another neuron through a synapse. Criteria for neurotransmitter. Nowadays, many substances are categorized as neurotransmitters. To consider a substance as a neurotransmitter, it should fulfill certain criteria as given below. It must be found in a neuron. It must be produced by a neuron. It must be released by a neuron. After release, it must act on a target area and produce some biological effect. After the action, it must be inactivated. Classification of neurotransmitters depending upon chemical nature. Many substances of different chemical nature are identified as neurotransmitters. Depending upon their chemical nature, neurotransmitters are classified into three groups. Amino acids. Neurotransmitters of this group are involved in fast synaptic transmission and are inhibitory and excitatory in action. GABA, glycine, glutamate, glutamic acid, and aspartate, aspartic acid, belong to this group. Amines. Amines are the modified amino acids. These neurotransmitters involve in slow synaptic transmission. These neurotransmitters are also inhibitory and excitatory in action. Noradrenaline, adrenaline, dopamine, serotonin, and histamine belong to this group. Others. Some neurotransmitters do not fit into any of these categories. One such substance is acetylcholine. It is formed from the choline and acetyl coenzyme A in the presence of the enzyme called choline acetyltransferase. Another substance included in this category is the soluble gas nitric oxide, NO. Depending upon function. Some of the neurotransmitters cause excitation of postsynaptic neuron while others cause inhibition. Thus, neurotransmitters are classified into two types: excitatory neurotransmitters, inhibitory neurotransmitters. 1. Excitatory neurotransmitters. Excitatory neurotransmitter is a chemical substance, which is responsible for the conduction of impulse from presynaptic neuron to postsynaptic neuron. Neurotransmitter released from the presynaptic axon terminal does not cause development of action potential in the postsynaptic neuron. Rather, it causes some change in the resting membrane potential, i.e. slight depolarization by the opening of sodium channels in the postsynaptic membrane and the influx of sodium ions from ECF. This slight depolarization is called excitatory postsynaptic potential, EPSP. EPSP in turn causes development of action potential in the initial segment of the axon of the postsynaptic neuron, chapter 140. Common excitatory neurotransmitters are acetylcholine and noradrenaline. Inhibitory neurotransmitters. Inhibitory neurotransmitter is a chemical substance, which inhibits the conduction of impulse from the presynaptic neuron to the postsynaptic neuron, chapter 140. When it is released from the presynaptic axon terminal due to the arrival of action potential, it causes opening of potassium channels in the postsynaptic membrane and efflux of potassium ions. This leads to hyperpolarization, which is called the inhibitory postsynaptic potential, IPSP. When IPSP is developed, the action potential is not generated in the postsynaptic neuron. Common inhibitory neurotransmitters are gamma-aminobutyric acid, GABA, and dopamine. Transport and release of neurotransmitter. Neurotransmitter is produced in the cell body of the neuron and is transported through axon. At the axon terminal, the neurotransmitter is stored in small packets called vesicles. Under the influence of a stimulus, these vesicles open and release the neurotransmitter into synaptic cleft. It binds to specific receptors on the surface of the postsynaptic cell. Receptors are G proteins, protein kinase or ligand-gated receptors. Inactivation of neurotransmitter. After the execution of the action, neurotransmitter is inactivated by 4 different mechanisms. It diffuses out of synaptic cleft to the area where it has no action. It is destroyed or disintegrated by specific enzymes. It is engulfed and removed by astrocytes, macrophages. It is removed by means of reuptake into the axon terminal. Reuptake of neurotransmitter. Reuptake is a process by which the neurotransmitter is taken back from synaptic cleft into the axon terminal after execution of its action. Reuptake process involves a specific carrier protein for each neurotransmitter. Important neurotransmitters. Some of the important neurotransmitters are described here. Details of neurotransmitters are given in tables 141.1 and 141.2. Acetylcholine. Acetylcholine is a cholinergic neurotransmitter. It possesses excitatory function. It produces the excitatory function by opening the ligand-gated sodium channels, chapters 32 and 140. Source. Acetylcholine is the transmitter substance at the neuromuscular junction and synapse. It is also released by the following nerve endings: preganglionic parasympathetic nerve, postganglionic parasympathetic nerve, preganglionic sympathetic nerve, postganglionic sympathetic cholinergic nerves, nerves supplying eccrine sweat glands, sympathetic vasodilator nerves in skeletal muscle, nerves in amacrine cells of retina. Many regions of brain. Synthesis. ACH is synthesized in the cholinergic nerve endings. Synthesis takes place in axoplasm and ACH is stored in the vesicles. It is synthesized from acetyl coenzyme A, acetyl-CoA. It combines with choline in the presence of the enzyme choline acetyltransferase to form ACH. Fate. Action of ACH is short-lived. Within 1 millisecond after the release from the vesicles, it is hydrolyzed into acetate and choline by the enzyme acetylcholine esterase, Fig 141.1. This enzyme is present in basal lamina of the synaptic cleft. Acetylcholine receptors. There are two types of receptors through which ACH acts on the tissues, namely, muscarinic receptors and nicotinic receptors. Reason for the terminology of these receptors is as follows: poisonous substance from toadstools called muscarine acts on a specific group of receptors known as muscarinic receptors. Similarly, another substance called nicotine acts on a specific group of receptors known as nicotinic receptors, but ACH acts on both the receptors. Muscarinic receptors are present in all the organs innervated by the postganglionic fibers of the parasympathetic system and by the sympathetic cholinergic nerves. Nicotinic receptors are present in the synapses between preganglionic and postganglionic neurons of both sympathetic and parasympathetic systems. Nicotinic receptors are also present in the neuromuscular junction on membrane of skeletal muscle. Noradrenaline. Noradrenaline is the neurotransmitter in adrenergic nerve fibers. It is released from the following structures: postganglionic sympathetic nerve endings, cerebral cortex, hypothalamus, basal ganglia, brainstem, locus ceruleus in pons, spinal cord. In many places, noradrenaline is the excitatory chemical mediator, and in very few places, it causes inhibition. It is believed to be involved in dreams, arousal, and elevation of moods. Refer Chapter 71 for the synthesis of noradrenaline. Dopamine. Dopamine is secreted by nerve endings in the following areas: basal ganglia, hypothalamus, limbic system, neocortex, retina. Small, intensely fluorescent cells in sympathetic ganglia. Dopamine possesses inhibitory action. Prolactin inhibitory hormone secreted by hypothalamus is considered to be dopamine. Refer Chapter 71 for the synthesis of dopamine. Serotonin. Serotonin is otherwise known as 5-hydroxytryptamine, 5-HT. It is synthesized from tryptophan by hydroxylation and decarboxylation. Large amount of serotonin, 90%, is found in enterochromatin cells of GI tract. Small amount is found in platelets and nervous system. It is secreted in the following structures: hypothalamus, limbic system, cerebellum, dorsal raphe nucleus of midbrain, spinal cord, retina, GI tract, lungs, platelets. It is an inhibitory substance. It inhibits impulses of pain sensation in posterior gray horn of spinal cord. It is supposed to cause depression of mood and sleep, chapter 145. Serotonin causes vasoconstriction, platelet aggregation, and smooth muscle contraction. It also controls food intake. Histamine. Histamine is secreted in nerve endings of hypothalamus, limbic cortex, and other parts of cerebral cortex. It is also secreted by gastric mucosa and mast cells. Histamine is an excitatory neurotransmitter. It is believed to play an important role in arousal mechanism. Gamma-aminobutyric acid. Gamma-aminobutyric acid, GABA, is an inhibitory neurotransmitter in synapses, particularly in CNS. It is responsible for presynaptic inhibition. It is secreted by nerve endings in the following structures: cerebral cortex, cerebellum, basal ganglia, spinal cord, retina. GABA causes synaptic inhibition by opening potassium channels and chloride channels. So, potassium comes out of synapse and chloride enters in, chapter 140. This leads to hyperpolarization, which is known as inhibitory postsynaptic potential, IPSP. Substance P. Substance P is a neuropeptide that acts as a neurotransmitter and as a neuromodulator, see below. Substance P is a polypeptide with 11 amino acid residues. It belongs to a family of 3 related peptides called neurokinins or tachykinins. The other peptides of this family are neurokinin A and neurokinin B, which are not well known like substance P. Substance P is secreted by the nerve endings, first-order neurons, of pain pathway in spinal cord. It is also found in many peripheral nerves, different parts of brain, particularly hypothalamus, retina and intestine, chapter 44. It mediates pain sensation. It is a potent vasodilator in CNS. It is responsible for regulation of anxiety, stress, mood disorders, neurotoxicity, nausea and vomiting. Nitric oxide. Nitric oxide, NO, is a neurotransmitter in the CNS. It is also the important neurotransmitter in the neuromuscular junctions between the inhibitory motor fibers of intrinsic nerve plexus and the smooth muscle fibers of GI tract. Nitric oxide acts as a mediator for the dilator effect of ACH on small arteries. In the smooth muscle fibers of arterioles, NO activates the enzyme guanylyl cyclase, which in turn causes formation of cyclic guanosine monophosphate, cGMP, from GMP. The cGMP is a smooth muscle relaxant and it causes dilatation of arteriolae. Thus, NO indirectly causes dilatation of arterioles. Peculiarity of NO is that it is neither produced by the neuronal cells nor stored in the vesicles. It is produced by non-neuronal cells like the endothelial cells of blood vessels. From the site of production, it diffuses into the neuronal and non-neuronal cells where it exerts its action. Neuromodulators. Definition. Neuromodulator is the chemical messenger, which modifies and regulates activities that take place during the synaptic transmission. These peptides do not propagate nerve impulses like neurotransmitters. Neuromodulators vs neurotransmitters. Neuromodulators are distinct from neurotransmitters. However, both the terms are wrongly interchanged. Neurotransmitters propagate nerve impulses through synapse, whereas neuromodulators modify and regulate the activities of synaptic transmission, Table 141.3. Neurotransmitters are packed in small vesicles in axon terminals only. But neuromodulators are generally packed in large synaptic vesicles, which are present in all parts of neuron like soma, dendrite, axon, and nerve endings. Many neurons have one conventional neurotransmitter and one or more neuromodulators. Few peptides like substance P, see above, act as neurotransmitters and neuromodulators. Actions of neuromodulators. Neurotransmitters affect the excitability of other neurons or other tissues, like muscle fiber, by producing depolarization or hyperpolarization through the receptors of ionic channels. But neuromodulators have diverse actions such as, regulation of synthesis, breakdown, or reuptake of neurotransmitter. Excitation or inhibition of membrane receptors by acting independently or together with neurotransmitter. Control of gene expression. Regulation of local blood flow. Promotion of synaptic formation. Control of glial cell morphology. Regulation of behavior. Chemistry of neuromodulators. Generally the neuromodulators are peptides. So neuromodulators are often referred as neuropeptides. Almost all the peptides found in nervous tissues are neuromodulators. Types of neuromodulators. Neuromodulators are classified into two types: non-opioid peptides. Opioid peptides. Non-opioid peptides. Non-opioid neuropeptides act by binding with G-protein coupled receptors. These neuropeptides are also called non-opioid neuromodulators. Non-opioid peptides are listed in Table 141.4. Opioid peptides. Peptides, which bind to opioid receptors, are called opioid peptides, Table 141.5. Opioid peptides are also called opioid neuropeptides or opioid neuromodulators. Opioid receptors are the membrane proteins located in nerve endings in brain and GI tract. Opioid receptors are of three types, and delta. These proteins are called opioid receptors because of their affinity towards the opiate or morphine, which are derived from opium. Opium is the juice of white poppy, Papaver somniferum. It is used as a narcotic to produce hallucinations and induce sleep. Opiate also induces sleep. Morphine is a powerful analgesic, pain reliever. Both opiate and morphine have high medicinal values, but are highly addictive. These two substances act by binding with the receptor proteins, opioid receptors, for the natural neuropeptides. Natural neuropeptides are called endogenous opioid peptides. Endogenous opioid peptides have opiate-like activity and inhibit the neurons in the brain involved in pain sensation. Opioid peptides are of three types: enkephalins, dynorphins, endorphins. Enkephalins. Enkephalins are the natural opiate peptides recognized first in pig's brain. Derived from the precursor proenkephalin, these peptides are present in the nerve endings in many parts of forebrain, substantia gelatinosa of brainstem, spinal cord, and GI tract. Two types of enkephalins are known: leucine enkephalin, YGGFL, and methionine enkephalin, YGGFM. Dynorphins. Dynorphins are derived from prodynorphin. Dynorphins are found in hypothalamus, posterior pituitary, and duodenum. Dynorphins are of two types, alpha and beta dynorphins. Endorphins. Endorphins are the large peptides derived from the precursor pro-opiomelanicordin. Endorphins are predominant in diencephalic region, particularly hypothalamus and anterior and intermediate lobes of pituitary gland. Three types of endorphins are recognized: alpha-, beta and gamma endorphins. Co-transmission and co-transmitters. Co-transmission is the release of many neurotransmitters from a single nerve terminal. Co-transmitters are the neurotransmitter substances that are released in addition to primary transmitter at the nerve endings. For many years, it was believed that each neuron releases only one neurotransmitter substance from its terminals. Now it is known that some of the neurons release many neurotransmitter substances. It is also believed that the additional neurotransmitters, i.e. the co-transmitters, modulate the effects of primary neurotransmitters. Some of the primary neurotransmitters act as co-transmitters in other nerve endings. Examples of co-transmitters: calcitonin, dopamine, dynorphin, GABA, gene-related peptide, glutamate, glycine, neuropeptide Y, substance P, vasoactive intestinal polypeptide, VIP.